Hello everyone. In this class, we'll briefly try to learn about how substances are transported across cell membrane. So this is a fluid mosaic model of the cell membrane where it is made up of a phospholipid bilayer and there are many integral proteins like peripheral proteins, the channel proteins and carrier proteins. And this membrane is permeable for highly lipid soluble substances like oxygen and carbon dioxide and nitrogen. And some of the water insoluble substances are they cannot diffuse through the bilipid membrane, so they take the help of channel proteins or carrier proteins for their transport. The transport across cell membrane occurs by two major processes, the active process and passive process. The passive process, here the substance move across the membrane without any energy expenditure, which is going to include diffusion and osmosis. So diffusion is nothing but it is a passive process by which molecules move from areas of higher concentration to areas of low concentration. So again, diffusion can be of two types. It is simple diffusion and facilitated diffusion. Simple diffusion occurs because of the heat content of the solution that keeps the solvent and the solute particles in the solution in a constant motion. The factors which are going to affect the diffusion are the distance. The greater the distance, the longer the time required. The size of the gradient, the larger the concentration gradient created, faster will be the diffusion. Temperature, the higher the temperature, faster the diffusion rate will be, and definitely depends on the molecular size. The permeability of the cell membrane to a substance falls rapidly with increase in molecular weight and in the large range between 10,000 to 60,000. So this is why glucose diffuses faster than large proteins. Then it depends on the lipid solubility. The lipid soluble molecules diffuse very rapidly, and of course it depends on the surface area. The larger surface area, faster the diffusion. The rate at which the material diffuses through the membrane is given by Fick's law of diffusion, which tells that the net rate of diffusion is directly proportional to the area of the cell membrane and the concentration of that particular substance inside and the outside, the difference of that particular substance inside and outside divided by the thickness of the cell membrane. So here where the C in concentration in and concentration out, they are nothing but the concentration of the material inside and outside the cell membrane. Facilitated diffusion. It is a carrier mediated process that enables molecules that are too large to flow through the membrane channels by simple diffusion. The carrier molecule undergoes repetitive spontaneous configurational changes during which the binding site for the substance is alternatively exposed to intracellular fluid and extracellular fluid. For example, glucose transport by the glucose transporter, that is GLUT, across the intestinal epithelium is a good example for facilitated diffusion. And transport of glucose into RBC, muscle and adipose tissue in the presence of insulin is also example for facilitated diffusion. Osmosis also kind of a simple diffusion. It refers to the diffusion of water molecule or water in any other solvent molecules through semi-permeable membrane. So this is the diagram showing how the substances are moved with help of carrier proteins. They are going to bind to the appropriate carrier proteins and there will be a configurational changes which helps in the movement of the substances through the carrier protein molecule. Coming to active process, here where the substances are transported against their chemical and electrical gradient. This form of transport requires energy in terms of ATP. So the different types of active transport includes are the primary active transport, secondary active transport, and vesicular transport. The primary active transport, they directly use energy from the hydrolysis of ATP. Some of the examples for primary active transport are sodium potassium pump, calcium pump, and potassium hydrogen pump. Sodium potassium pump. So it is an electrogenic pump. Here 3 sodium ions moves out and 2 ions moves in. It uses ATPase as carrier molecule, and ATPase is comprised of 6 subunits, 3 alpha and 3 beta subunits. Sodium and potassium transport occurs through alpha subunit. The alpha subunit has ATPase enzymatic activity too, and binding sites are present on its intracellular and extracellular phases. The mechanism of action of sodium potassium pump. The binding of 3 sodium ions and ATP to a carrier protein inside the cell transfers high energy phosphate group from ATP to aspartic acid residue of alpha subunit ATPase, so which leads to phosphorylation. So this causes change in the configuration of protein, resulting in 3 sodium ions to move out of the cell. And when 2 potassium ions bind to carrier protein on the outside the cell, the aspartic acid phosphate bond is hydrolyzed, so which leads to dephosphorylation. This causes the second change in the configuration of protein, resulting 2 potassium ions to move into the cell. So this is the diagram of sodium potassium pump, where you can see the alpha subunit and beta subunit. Okay, so most of the receptors for sodium and potassium, they are present on the alpha subunit inside and outside, whereas it is also binding site for ATP intracellularly. So when 3 sodium and ATP binds, okay, there will be a transfer of energy which causes the configurational changes in the alpha protein, which moves the sodium out of the cell. So that leads to binding of potassium, 2 potassium ions from the outside the cell, and so this causes the dephosphorylation, which is going to lead to the configurational changes in the alpha subunit again, so which helps in the movement of 2 potassium inside the cell. Okay, so there is a net loss of 1 positive ion inside the cell, and that is how the sodium potassium pump increases the negativity inside the cell. Okay, and with the net loss of 1 positive ion or sodium ion, so there will be movement of the water also from intracellular to extracellular fluid. Okay, and that is how the sodium potassium pump, which also help in the regulation of the volume of the cell. And another function is because of the ATPase activity, the heat which is going to released, it is very important for the cellular functions. Clinical significance. Digitalis, a drug used for the treatment of heart failure, increases the myocardial contractility, binding to alpha subunit and interfering with the dephosphorylation step of transport process. This is the prominent in failing heart. Calcium pump. It is present in sarcoplasmic reticulum of muscle to maintain intracellular calcium concentration. It is also present in cell membrane and cell organelle membrane. In the cell membrane, the direction of calcium is from cytoplasm to extracellular fluid. In the cell organelle members, it is from cytoplasm to the organelle lumen. The third example is potassium hydrogen pump. It is present in cells of gastric mucosa and renal tubules, where it causes active secretion of H+ ion. Some of the substances which can block these channels, the sodium channels are blocked by tetrodotoxin and saxitoxin, whereas potassium channels are blocked by tetraethyl ammonium. Calcium channels can be blocked by verapamil and nifedipine, whereas sodium potassium ATPase can be uh, blocked by obin and digitoxin. Coming to secondary active transport, here the active transport of sodium is coupled with the transport of other substances. Mechanism: when both sodium and the substance are bound to the carrier molecule, the carrier undergoes configurational change during which both these molecules are transported across the cell membrane. For example, the co-transport of glucose and amino acids along with sodium from the proximal renal tubules. Another example is sodium countertransport of calcium and H plus ion, uh, H plus ion, where the transport is in direction opposite to the primary ion, that is sodium. So this is the example for uh, the secondary uh, active transport, where the sodium, when it moves, okay, so it is going to move with another molecule like glucose or amino acid, so which is going to lead to the co-transport. Okay. So the movement of sodium occurs mainly because of the concentration gradient created for the sodium because of the sodium potassium pump activity at the basolateral membrane, so which is going to pump 3 sodium ions out and brings 2 potassium inside. Okay. So that is going to create uh, the the concentration gradient for the sodium. Okay, so that is why the sodium is going to move from the extracellular fluid to intracellular fluid. With that, the another substance is going to move. Okay, so which is called as secondary active transport. So it is called as secondary active transport because the concentration gradient for sodium is created because of the primary active transport here. Coming to vesicular transport, so which occurs by endocytosis and exocytosis. Endocytosis, there are two types. One is phagocytosis and another one is receptor mediated endocytosis. Phagocytosis is a process by which the extracellular substances, that is bacteria, dead tissue, foreign particles are engulfed by the cells. The substance make contact with the cell membrane, which then invaginates. The endocytic vesicle pinches off from the cell membrane and fuse with another intracellular vesicle like lysosome, from which the ingested substance is released into the intracellular fluid. There can be receptor mediated endocytosis, where the material to be transported first bind to the receptor and then receptor substance complex is ingested by endocytosis. Now, the example for receptor mediated endocytosis is clathrin mediated endocytosis. Exocytosis occurs when substance secreted by the cell are trapped within the vesicles or granules, which fuse with the membrane and then release their contents into the extracellular fluid. Okay, so here some of the substances which are secreted by the cell itself, they are trapped into the vesicle. Then these vesicle fuse with the plasma membrane and then there will be exocytosis. Okay, and if the endocytosis can occur at one side and if exocytosis can occur on the other side of the cell membrane, so then there can be actual transfer of molecules from one side to another side of the cell, so which is called as transcellular transport. Carrier type process. The carrier is a protein that binds ion and other molecules and then change their configuration, thus moving the bound or molecule from one side of the membrane to other. There are different types of carrier proteins. They are unipotent, where the transport of a single particle in one direction occurs. For example, facilitated diffusion of glucose. Sympotous, where two particles together in the same direction are transported. For example, in secondary reactor transport of glucose. In the antipoders, the transport of molecules occurs in opposite direction. For example, sodium potassium pump, sodium calcium exchanges, and sodium hydrogen exchanges. Okay. So these are the example showing the uniport mechanism and the coupled transport, so which can be symport or antiport. In symport, the two substances, they are going to move in a similar same direction, whereas in anti-transport, the substances are going to move in opposite direction. So this is in brief about the transport of substances across the cell membrane. Thank you.